This video is about the timing and coordination of development. When you were very first conceived, you began as a zygote, and through a very precise set of steps regulated by genes and instigated by other environmental factors, you developed from a zygote to an embryo to a fetus, and then were a complete organism with fully functioning organs, organ systems, and tissues of all different kinds of cells. The big ideas for this video are that timing and coordination of specific events are necessary for the normal development of an organism, and that these events are regulated by a variety of mechanisms. Also, that interactions between external stimuli and regulated gene expression result in the specialization of cells, tissues, and organs. There are four processes that underlie development: determination, differentiation, morphogenesis, and growth. And so you begin as a fertilized egg, and that's called a zygote. Then that zygote will start to divide into a ball of dense ball cells called a morula. Then division will continue, but then the ball will kind of hollow out into what's called a blastocyst. So this picture is showing a section of a blastocyst where determination is occurring. So there are three cell layers that start to separate in about the second week of development. And it's the ectoderm, from which all the nervous tissue is going to derive, the mesoderm, which will give us our muscle, our connective tissue, blood cells, etc., and then endoderm, which is going to give you things like your pancreas and your thyroid. So this is the first step in this determination, and going from cells that could turn into anything, and now we're headed down the road to specialization. After determination is differentiation, and so this is even further specialization. It results from the expression of genes for tissue-specific proteins, right? Because eyeballs don't need to be producing proteins to make toenails, and hair follicles certainly don't need to be making milk, like mammary glands would. So this differentiation, we've got different genes expressing the proteins for that cell type. And here's an example of how we might know that. So a scientist took a slice of an embryo and put this set of undifferentiated cells in a petri dish. When the set of undifferentiated cells was exposed to about 100 micrograms per milliliter of a protein called activin, it became differentiated into heart and cartilage tissue. If you took the same undifferentiated cells and exposed it to a much smaller dose of that same protein, it would differentiate into muscle cell. And if you combined that protein with one called RA in a different concentration, then you would start to develop tissue. This is called a pronephros, which is going to be the pregame to kidneys. So differentiation, we're going to get that from genes making proteins that are tissue specific. The next step is morphogenesis. So our cell types have been determined, they've been differentiated, and now morph means shape, genesis means beginning. So this is when our organism are really starting to kind of mold it and have it take shape. Cell division is part of this, cell expansion and movement, and even apoptosis, which is programmed cell death. And as we know, you think death, how is that supposed to help me grow and develop into an organism? Well, here's an example. When you're first developing, your hands are kind of like little paddles, your hands and your feet, and then there comes a time when your bones and all those things start to fully develop, that apoptosis happens in between your digits so that you have fully separated hands. And here's some examples when it didn't quite work all the way out, and there's still some of that webbing because apoptosis did not completely happen. There are genes that regulate this morphogenesis. And one set of those is homeotic genes. And homeotic genes are sets of genes, like this is a set, this is a set, this is a set, right? That regulate the fate of body segments. So whether you're going to be near the head or near the arms or near the middle or down at the end of the abdomen, right? So they regulate the fate of the body segments by regulating the expression of those tissue-specific genes. And what's interesting is that this is Drosophila melanogaster, fruit flies, and then here is us, Homo sapiens humans, and we have a lot of the exact same genes and types of genes in the same order, in the same places as fruit flies do. So, and they do the same job. So that's kind of cool because that's a homologous, these are homologous genes, just like homologous structures, right? They're similarities that indicate a common ancestry. So the fact that we even have homeotic genes is evidence of common ancestry. Another thing that we have, another set of genes and molecules that help differentiate and actually will shape the body of an organism, are morphogens. And these are signaling molecules whose differential concentration, that just means there's more in some place than in other places, so whose differential concentration gradient influences patterns of development. So an example here is a Sonic the Hedgehog gene. Yes, that is a real name of a real gene, and Sonic Hedgehog protein. And its job is to help your limbs figure out, is it thumb side of the hand or pinky side of the hand, so that as your fingers grow, otherwise you have a thumb on this side and all kinds of weird action. So it helps you know, like, this is the midline of the body, and these are the sides, and this is the front, and this is the back. And the amount of genes in these, the amount of the protein in each of these locations helps direct the morphogenesis. Now what's interesting is if that doesn't quite work out like it's supposed to, nature comes up with all kinds of interesting things. Right? So like, here's a two-headed pig, and here's a cyclops baby shark, and here are some limbs, some forelimbs and hind limbs with more or misshapen digits. So if there's too much or not enough, or if Sonic Hedgehog is being expressed in places where it's not supposed to, very interesting things can happen. And the final part of development is growth, and that's just where we go from our, we're all differentiated, and now it's time to grow up. So we're getting all the things we need, right? The right amount of food, the right amount of water and nutrients, the right amount of energy, and hormones like human growth hormone, stimulating cell division, and the cell cycle. So that's it for people. Let's take a look at plants briefly. They just go through a very similar thing. They go through determination from the very beginning, from the very first division of the cell, of the zygote. Determination has occurred. This apical cell, or the cell at the top, is going to be the mother cell of all the shoots and leaves, and the basal cell is going to be the progenitor of all the cells that will make up the root tissue. So you can see, right, you had a zygote, then there's differential gene expression, so transcriptional activation, right, that's some gene expression of these genes here, and then environmental cues like presence of water or presence of hormones like gibberellins and auxins will stimulate germination. You can see here, again, there was that determination. Then there starts to be differentiation, right? So going from just two, either apical or basal, into these 4 different tissue types, and then even further, then we get into morphogenesis, and we're starting to see the actual cotyledons or baby leaves and root take shape. And again, this is just showing us about how timing and coordination of specific events are necessary for development, and that they're regulated by many different kinds of mechanisms, and that the interactions between the external stimuli and that regulated gene expression can result in specialization of cells and tissues and organs.